So in 2006, uh, in Uganda, centralized mode of testing was rolled out, uh, basically to scale up early infant diagnostics testing across the whole of Uganda. So currently, over 1.1 million samples are received annually at central public health laboratories. And of these, approximately 80,000 samples are received monthly from the HIV viral load testing program, and approximately 12,000 samples from the early infant diagnosis program monthly. And some of the other samples we receive are like the microbiology isolates from the antimicrobial drug resistance surveillance and outbreak investigations, sickle cell DBS samples, and hepatitis B samples, among others. For the centralized testing to be implemented, a national sample transport network was set up across the whole country, and it is accessible to approximately 2,800 facilities. And there are a total of 100 functional hubs across the whole country. And these hubs act like coordination centers to approximately 20 to 30 health facilities. So how this is done is samples are picked from the remote health facilities, uh, and they are transported to the hub. And they are transported by motorbikes on and bike riders. And these samples are picked by motor vehicles and motorbikes from the hubs and transported to the central testing lab, which is Central Public Health Laboratories. So this centralized testing model has enabled us to get huge amounts of samples from across the country that are representative of the whole population. So being able to acquire all these samples at the Central Public Health Laboratories were initially challenged with the incorrect storage of this biospecimen, and this was leading to inaccessibility and difficult retrieval upon request by researchers. So in our process to try to correctly store these samples for them to be retrievable, we expressed that we lack informed consent from the patient donors to allow for storage and utilization of this biospecimen. And through the consultations with the regulatory authorities and also some of the consultants on the subject matter, we realized that there is unregulated biospecimen storage and unmonitored biorepositories in Uganda, which is of a very big biosecurity concern. And there is also absence of a functional repository in a context that the biorepositories that exist in Uganda have a very narrow scope. They are looking at specific research interests. So when you add all this together, there is a huge extent of export of biospecimen, and it is increasingly increasing missed opportunities to all the patient donors, the research institutions in Uganda, and also as a country as a whole. So that is when we established the National Biorepository, and we are aiming at appropriately storing this biospecimen and seeking informed consent for the storage of this biospecimen. We are aiming at fostering international and local collaborations and building capacity in biobanking science as a whole. So our first step in planning and development was to mobilize for resources and also infrastructure development. We set apart a space for the National Biorepository and also procured some storage facilities. And we also tried to review the organization structure to appoint the key positions for the biorepository. And the next step was to set up a task force which wrote the National Biorepository proposal. And also standard operating procedures were written out. And these SOPs were written in line with the International Society for Biological and Environmental Repositories best practices, which has helped us in the guidance of the SOPs. All these documents were submitted to the Research Ethics Committees and Uganda National Council for Science and Technology. And Uganda National Council for Science and Technology is like the regulatory body responsible for all research that is using humans as research participants. And however, despite the engagement with UNCST, the approvals are still pending up to now. And because of this is where most of the regulatory and ethical uncertainties are still of challenge in our country. So what we are advocating for is for education in biobanking science and biobanking ethics in low and middle income countries, because of this inadequate specialized biobanking knowledge that is the major cause of this lack of guidelines. And then the regulatory authorities should generate country-specific biobanking policies and guidelines, and they should really do this in engagement to the key stakeholders. And the question we would want to ask us as developing countries, to what extent should we engage the stakeholders when it comes to the concept of writing policies and guidelines? You need to involve the lawmakers, because you find that most of these policies have to be backed up by the law, and also by repository managers and institutional officials, because of the wide scope of biorepositories that are in different institutions. This might be a veterinary biorepository, a microorganisms biorepository. So the policies should be able to address all these and should be contextual in our setting. And mid this year, we conducted what we called a stakeholders consultation meeting. And what we did was to engage the regulatory bodies, the lawmakers, the health and academia, and also the development partners. So the objectives of this meeting wanted to understand how to get valid informed consent for the biospecimens that are already in the storage in the National Biorepository. And we wanted to understand what is the role of the Research Ethics Committee and the regulatory bodies when it comes to establishment of biobanks in Uganda and also biospecimen research. And we also use this platform to network with research institutions and academia on how to promote biospecimen utilization. And it was a fruitful discussion, and some of the ongoing activities that were generated from the stakeholders meeting was to review the laboratory sample request form and to incorporate the informed consent statement for storage and future use of these samples. And for the samples that are already in storage in the National Biorepository, they advise us to seek legal opinion through the Attorney General. So that is what we are trying to fast track. And the regulatory body, which is Uganda National Council for Science and Technology, was also tasked to compile the biobanking guidelines and policies that are in line with international standards. Just to take us through something that we all know is informed consent. So the UNCST guides us that a specific informed consent form should be used for samples collected with an intention of being stored and used for future studies. So informed in such a way that the patient donor should have sufficient knowledge and understanding about the sample that they are donating, and they should decide voluntarily whether to accept or or deny. They should provide this permission. And this process is truly, it should be ongoing to the context of the patient donor. So this model, according to the guidelines, offers the best protection for autonomy. However, it has some limitations on our end. The intended future use of the biospecimen and associated data are unknown at the time of diagnostic testing. And this is being explained in a context of clinical setting or a health program, like the way that is applicable to the National Biorepository. And then secondly, the high workloads that might affect specific informed consent. And in a clinical care setting, they consider informed consent as an added role. Like, we can just allow me to compare, like in a research setup, you have these predefined visits. You are aware of how many patients are coming in a specific day. But in a healthcare setting, the visits are not predefined. The patients come in to seek healthcare. So these healthcare workers will naturally prioritize the offering of primary healthcare and consider the informed consent process later. So the idea here is to try to think through in a context of the National Biorepository, what is the informed consent alternatives that we should think about. The first one is the opt-out principle. This should be either regulation or a law, and obligatory information is offered to the patient donor at the point of sample collection. And at that point, consent for storage of the leftover sample is presumed to be given. However, procedures need to be put in place for the patient donor to withdraw at any one point when they see fit. The other alternative is the broad consent, and this is where unspecified information about the future and possible uses of the research using this sample is shared with the patient donor. And they still, with this information, they decide to either accept or deny, and still processes need to be put in place to opt out. So through just the brief overview of our ethical uncertainties, I would want to just really hear your submissions on these points. What type of informed consent is appropriate for a biobank that targets storage of remnant clinical biospecimen, and how best may we overcome the apparent obstacle of lack of prior informed consent for the biospecimen stored in the National Biorepository? And would want to also get to understand some of the useful models, to share some of the useful models and best practices of informed consent templates that may be applicable to us. I would like to acknowledge our partner institutions who have supported us through the work that is being done in the National Biorepository. Thank you.